First part, we're going to start talking about antibiotics and what do you use them for. So the first thing to remember is anti or against. So okay, anti means against. And don't forget, you're also in biology class. So in this class we study living things or biology. So biology equals living things. Now, an antibiotic is basically something that's going to kill something that's living, okay? Now, my major example is you're not going to use an antibiotic to kill an elephant. An elephant is one of the largest land mammals on Earth, and you're not going to give it a couple small pills if you're trying to kill it. Okay, we are going to use antibiotics to kill what are called microorganisms, the very smallest creatures on Earth. Okay, so bacteria, those are the microorganisms. Micro meaning small, and organism, something that's living. Remember, when we were doing classification, microorganisms are going to be put into three different classes. There's regular bacteria, archaea bacteria. These are some of the most primitive. They're going to be found in really cold environments or really hot environments, and they're the most simplest bacteria. And then remember, eubacteria. Eubacteria, the way is you can remember those are more complex, and they're the ones that make you or me sick. We use the antibiotics to fight these living things. An antibiotic is basically fungus, a mold. And we found, starting in the 1930s, that mold can kill small microorganisms. So moving on, we have, as a society, have developed different antibiotics to fight bacterial infection. Now remember, there's bacterial infections and there's viral infection. The virus would, example would be influenza, cold, chickenpox. Examples of bacterial infections that are very well known include flesh-eating bacteria. That's what Jim Henson Productions, founder and the Muppets, passed away from. Strep throat, a lot of people get that. Staph infections, E. coli, and then one of the STDs that's known for it is gonorrhea. If you have a bacterial infection, doctors are going to want to know what type of bacteria it is. Bacteria are classified in two different ways. One is called gram-positive, and the other one is called gram-negative. They will use a stain in order to tell which one is which. If it is a gram-positive, they will stain it and it'll turn purple. If it is gram-negative, it'll turn pink. Okay, gram-positive cells are known for having a thick cell wall. Gram-negative ones are known for having a thin cell wall. Depending on the ability of the antibiotic to fight it, some will work better on a thick cell wall, and some will work better on a thin cell wall. And basically that's going to involve the ability of the antibiotic to penetrate the cell. There's two different ways in which they can stop a cell from developing into another generation. First one involves basically shortening or thinning out the cell wall. So in that case, there's different antibiotics that are specific to gram-positive cells. Then there's another one which basically stop the proteins in the cell. Easiest example I can tell you is we are all made of protein. We have skin, we have nails, we have teeth. Those are all made out of protein. If there was something that they could do to limit the ability of our bodies to make new proteins, we would slow down the synthesis or the making of them and then eventually die. If you have a cut on yourself and you're bleeding, your blood will clot and basically prevent the blood from falling out of you. Okay. If our bodies didn't have that ability to make the proteins in the blood to clot, we would eventually die. So think of that with the bacteria. If you can slow down the ability of the bacteria to redevelop and make new generations, you're limiting its ability to spread. So first one, we'll target the cell and its thickness and try to thin it. The other one will stop or try to slow down the development of protein. Now, the gram-negative ones that are actually the ones that thin out, actually the ones that concentrate on the protein synthesis. The proteins are developed in a different part of the ribosome in the bacteria versus humans, so that is one reason why they don't impact us, okay? That is one reason why we don't just stop. Now, during this lab, we are taking petri dishes, and they have E. coli on them. So E. coli is basically a common strain of bacteria. You can find it in meat. So one thing you might have heard is some form of E. coli poisoning. If you've ever had undercooked chicken, that usually because of E. coli poisoning. Now, the strain that we're using here is not going to harm us, but we are taking extra precautions and we're using gloves and protection. Now, you can fight an E. coli poisoning or infection using different bacterias. Now, we will coordinate off our sample into 4 different sections. One will be the control, and then there will be 1, 2, and 3. Those are the 3 different types of antibiotics that we're using to test. Now, the antibiotic is going to be on a little circular disc that you should have read about in the reading, and that's going to be touching the agar or the E. coli. We're going to come back after a couple days and check the effectiveness of that. If there are only a couple dots in like a quadrant here, or 1, 2, or 3, it's probably not really working. These are mutant cells or just outliers, okay? But nothing's really working. If we get a big circular disc that's different than the agar or the clear jelly, we know it's really working, because basically that's killing the bacteria that's in that section. And then in the medium part, if we see like a medium circle, maybe about half of the bacteria are dying from it, the other half are still living. So it's not quite as effective of an antibiotic. The reason why scientists use this is if you have a different type of infection, they want to see what kind of antibiotic works on that particular infection. So if they get a big circle and a good reaction from it, they know we should give this to that person. It'll fight off that infection. If they only get a couple dots in the sample, it's not effective. If it's a medium one, they could try that one, but there's probably a better antibiotic that'll work, but depending on the different type of infection, they might be limited to this, okay? Because not all, I mean, if you have like an allergy, like some people are really allergic to amoxicillin, you might not be able to use the particular antibiotic that really works. You might get stuck with this one, okay? So that's just a little overview of the lab, and if you have any questions, please let us know.